Insulin, a drug that every pharmacy student study, a drug that every diabetic patient depends on. But do you know the real story behind it? Do you know about the young surgeon who had a dream that changed everything? Do you know about the 14-year-old dying boy who became the first human to ever receive insulin? To understand why insulin was such a miracle, you first need to understand what diabetes meant before 1922. So, diabetes has been recognized as a disease for over 3 million. Patients who were extremely thirsty urinated frequently and withered away to nothing regardless of how much they ate were described by ancient doctors. And because diabetic patients' urine was sweet with sugar, ancient Indian doctors actually observed that it attracted ants, and they referred to it as honey urine. However, understanding the symptoms did not equate to understanding the remedy. A starvation diet was the only cure to for the diabetes in the beginning of 20th century. Patients were placed on diets with no carb and nearly no sugar, sometimes as low as 300 to 500 calories per day. They nearly survived as a result of this. They were skeletons on food. In an effort to keep their blood sugar levels low, children with diabetes were effectively starved to death. And one such youngster was Leonard Thompson. He was a youngster. He was born in Toronto, Canada in 1908, and he was given a diabetes diagnosis when he was 11 years old. He was barely 65 pounds, and when he was 14 years old, after being admitted to Toronto General Hospital, he was only given 450 calories a day while in and out of a diabetic coma. This was the ward into which Frederick Banting was about to make his entrance. Oscar Minkowski and Joseph von Merck, two German researchers, found in 1889 that dogs that had their pancreas removed developed symptoms of diabetes and soon died. This discovery gave rise to the theory that pancreatic substances, later identified as insulin, were created in the pancreas. Further research focused on pancreatic islets, which are collections of specialized cells. Sir Edward Albert Schaffer proposed in 1910 that individuals with diabetes had a single chemical deficiency in their pancreas. He gave this substance the name insulin, which comes from the Latin word insula and which means island. So Frederick Banting read an article from a medical publication in October 1920 while preparing a talk on the pancreatic. It implied that islets of Langerhans, the pancreatic cells that produce insulin, degrade more slowly than the other pancreatic tissue. Banting's breakthrough ideas came from this single line. And for many years, researchers had known that the pancreas secreted insulin, an endogenous hormone that regulated blood sugar. However, every attempt to extract it was unsuccessful because as soon as the gland was crushed, the insulin was instantly destroyed by the potent digestive enzyme of pancreas. So Banting's concept was straightforward. What if you tied up a dog's pancreatic duct first? The islet cells that produce insulin would endure, but the digesting portion of the pancreas would shrink and perish. Then, without being broken down by digestive enzymes, insulin could be taken from these surviving cells. What happened was next was ultimately a truly miraculous. Frederick Banting, a young surgeon, and Charles Best, his assistant, were able to isolate a pancreatic extract in 1921 that significantly reduced blood sugar level in dogs with diabetes. It was referred to as iletin. A dog that with severe diabetes was kept alive for 70 days by Banting and Best's murky mixture, and the dog only passed away when the extract ran out. Millions of people with diabetes now have hope thanks to this discovery. Building on his achievement, Banting and Best created a more pure and refined type of insulin, this time derived from the pancreases of cattle with assistance from J.B. Collett and John McLeod. The first person to receive an insulin injection was a 14-year-old Leonard Thompson, who was dying of diabetes in Toronto Hospital in January 1922. The first clinical experiment, however, was a failure. A 15 ml doses of pancreatic extract created a sterile abscess, had little effect on ketoacidosis, and only marginally decreased blood sugar and urine sugar. Because the extract was insufficiently pure, Leonard experienced a strong allergic reaction and his condition did not get better. So Colip spent 12 days working non-stop to further refine the insulin extract. And Leonard was given another round of shots on January 23rd. His blood sugar, urine sugar, and ketones all went back to normal this time. And in particular, his urine sugar decreased from 71 to 9 grams. His blood sugar dropped from 520 mg per dl to 120 mg per dl. And his ketonuria disappeared. Globally, word of this discovery spread like wildfire. Together with Best and Collip, Banting and MacLeod received the 1923 Nobel Prize in Medicine. Eli Lilly, a medical company, began producing insulin on a large scale shortly after. Before long, there was enough insulin to cover the whole continent of North America. Manufacturers created a number of slower acting insulin in the decades that followed. Novo Nordisk Pharmaceuticals released the first one in 1936. The pancreas of pigs and cattle was used to extract the insulin that saved Leonard Thompson in 1922. Early patients frequently experienced adverse reactions since it was animal insulin, which is different from human insulin. So, the development of recombinant human insulin required years of pharmaceutical study. And Eli Lilly created Humulin, the first biosynthetic human insulin in 1982, utilizing recombinant DNA technology. This insulin, which was created by introducing the human insulin gene into bacteria, had precisely the same amino acid sequence as a human insulin. And we have insulin analogs now, a century after Benting and Best, molecules whose amino acid sequence has been purposefully altered to have particular pharmacokinetic characteristics. Insulin is currently available in a variety of forms, ranging from ultra rapid and ultra long acting insulins to conventional human insulin, which is identical to what the body makes on its own. People with diabetes can select from a range of insulin formula and methods based on their individual needs and lifestyle, thanks to decades of study. It literally saved lives, even though it might not be a cure for disease. So, this is the story of insulin. If you found this story fascinating, share it with someone who takes insulin daily or knows someone who does. They deserve to know the story.